SV40. In order to understand SV40, meaning Simian Virus 40, we must discuss the past history of AIDS and inoculations. In 1969, Dr. William Campbell Douglas admitted that the AIDS virus was deliberately manufactured by the National Cancer Institute in collaboration with the World Health Organization, WHO, based on the Bulletin of the WHO, Volume 47, page 259, 1972. The U.S. Department of Defense sought funding to create a "synthetic biological agent," an agent that does not naturally exist and for which no natural immunity could have been acquired. From DoD, July, 1969. It was Dr. Theodore Strecker whose research proposes that the AIDS virus can be created by combining two deadly retroviruses, bovine leukemia virus, BLV, and sheep visna virus. Taken from Miller, page 50. Dr. E. L. Snead discovered that the Simian Virus 40 was associated with much research and interest in the carrier, the African green macaque monkey. Finally, in 1964, Horvath and Finosi published their findings stated in summary, SV40 virus has been recovered from 10 out of 35 9 to 12 months old babies primo-vaccinated orally with type 1 attenuated polyovirus vaccine contaminated with SV40 virus. It was found the excretors of SV40 developed a lower antibody level against type 1 polyovirus than non-excretors. Dr. Snead reminds us that children were screened for SV40 presence before immunization, as reported by Dr. Howard E. Gentleman, et al., published in the Proceedings of the National Academy of Sciences of the United States of America, Volume 83, pages 9759 through 9763, December 1986. SV40 has been shown to be one of the activators of the HIV or AIDS virus. Dr. Snead reports, since SV40 was present, yet passed undetected in the early specimens of Salk and Sabine vaccine, viruses other than SV40 could also have fooled us. The Science Digest published an article titled Near Disaster with the Salk Vaccine, December 1963, pages 40-41. The author, Arthur J. Snyder, states, "It is now almost certain that a recently discovered virus, unwittingly put into hundreds of thousands, if not millions, of doses of early Salk vaccine, will not cause cancer in human beings. There was also some evidence the SV40 virus could produce slight, non-cancerous infection in man, and when put in human tissue could cause some cell changes suggestive of tumor growth." Snyder reveals further when he states, "There is a slight increase in leukemia, but the same increase is shown in states receiving no SV40 in the vaccine." It means, there were records of which states received SV40 in the Salk-Sabine vaccines. Tissues infected with SV40 do not exhibit the usual decreases of DNA synthesis, and furthermore, protein synthesis does not necessarily copy or replicate the specific viral protein, again manifesting the unusual ability of this unique particle to mask, hide and deceive." Research of Snead from the Tadero study. This knowledge about SV40 was understood specifically by 1968. Review History of AIDS, 1959. World Health Organization issues standards for polio vaccine. The precautionary measures listed avoiding using simians monkeys already employed for other experiments. 1960. WHO issues bulletin about unexpected viruses that might be encountered in vaccines. 1961. Live virus vaccination was started and presumed safe. 1963. Journal of New York Academy of Science reported that humans were susceptible to a simian tumor virus. 1963. Science Digest explains the near disaster of polio immunization program. 1964-65. Excretion of SV40 virus after oral administration of contaminated polio vaccine. End quote from Horvath Research. 1977. February issue of Atlantic Monthly affirms the contamination of millions of Americans with SV40. 1977. First documented AIDS patient reported in Africa. Dr. Robert B. Strecker, in his Strecker Memorandum, stated AIDS is a man-made disease, planned specifically for testing in Africa. He documents that brain cancer is the result of the SV40 virus. Quotes, as a result of contaminated polio vaccine in 1960s, a polio-like disease from contaminated swine flu vaccine in 1970s, and AIDS from smallpox and hepatitis B vaccines. End quote. The Strecker Group, 1216 Wilshire Boulevard, Los Angeles, Car 90017. Dr. Stewart, et al., found, quote, according to these statistics, deaths, the risk of dying from a malignant disease after the age 40 has barely altered in recent years, from 1953 through 1955, but the risk of children and young adults dying from these diseases has appreciably increased. In particular, children between 2 and 4 years of age have been more affected by the unfavorable trend of leukemia mortality than any other age under 70 years. End quote. In 1960, the World Health Organization, WHO, issued a bulletin informing the public about unexpected, undesirable viruses that might be encountered in vaccines. In 1960, scientist Bernice Eddy discovered that rhesus monkey kidney cells used in the Salk polio vaccine and experimental oral polio vaccines could cause cancer when injected into lab animals. Later that year, the cancer-causing virus in the rhesus monkey kidney cells was identified as SV40 or Simian Virus 40, the 40th monkey virus to be discovered. Between 1955 through 1963, nearly 100 million American children had been given polio vaccine contaminated with the monkey virus, SV40. The American public found out February 1977 in the issue of the Atlantic Monthly that there was contamination of millions of Americans with SV40 through vaccinations. Finally, in 1977, the first official report of AIDS was reported in a Northern European female doctor in Africa. The 1997 National Institute of Health (NIH) conference simply could not agree on whether the SV40 was actually being identified in the cancerous tumors of children and adults alive today, and, if it was, whether the monkey virus was in fact responsible for their cancer. Scientists continue to identify SV40 in human brain and lung cancers of children and adults and are finding SV40 associated with bone cancers and non-Hodgkin's lymphomas. In a report published in 2001, the U.S. Institute of Medicine Immunization Review Committee stated, quote, the biological evidence supporting the theory that SV40 contamination of polio vaccines could contribute to human cancers. End quote. Author unknown. After 1963, the oral polio vaccine was made using the African green monkey. While the African green monkey is generally free of SV40 virus, it does carry a number of viruses, including Simian Virus, SCMV. The polio vaccine developed by Dr. Salk was not safe because the 1 in 4,000 dilution of formaldehyde, which barely inactivated the poliovirus, did not fully inactivate SV40. The Vilches study showed that poliovirus SV40 T antigen DNA sequences are significantly associated with non-Hodgkin lymphoma in HIV-1 infected and HIV-1 uninfected patients. Quote, half of the 55,000 cases of non-Hodgkin lymphoma in the U.S. are likely related to contaminated polio vaccine. End quote. Taken from Merkel Research. The major oral polio vaccine manufacturer did not adequately test their master seed stocks, which reportedly contained SV40, and used them to produce vaccine released for use to American children from 1963 to the 1990s. Since 1999, the US has discontinued the use of live oral polio vaccines, and children receive a killed polio vaccine that is, hopefully, SV40 free. Two studies, Shiva-Perka and Vilshay, have found a relationship between SV40 and non-Hodgkin's lymphomas. Even though Boutel and other studies insist there is no cancer relationship, the public should seek further information. Ironically, there are no studies to show the extent of the presence of SV40, or if there is an increase of SV40 in the world populations. Neither is there mainstream concern about SV40 being an epidemic. Quote, "It has been noted that different strains of SV40 may not be readily identified by in vitro culture methods." End quote from the European agency. Carbone found, quote, "SV40 has been linked to some human malignancies, and the evidence that this virus plays a causative role in mesothelioma and brain tumors is mounting." End quote. Heinonen et al. 1973, reported a higher incidence of neural malignancies in children born to mothers who received inactivated poliovirus during pregnancy. Boutel and colleagues found genetic sequences from SV40 in 42% of the 154 non-Hodgkin lymphomas they analyzed. 3 of those non-Hodgkin samples contained strains identical to that in the 1955 polio vaccine. Epidemiologist Howard Strickler of Albert Einstein College of Medicine in the Bronx, New York, stated, quote, "Certainly, I think the evidence is there, that SV40 or fragments of SV40 are present in a wide variety of tumors." End quote. Strickler further stated, quote, "Mesothelioma was primarily linked to the exposure of asbestos. Studies have found that around 70% of mesothelioma cases test positive for the SV40 virus. Over the past 30 years, the number of mesothelioma cases has risen tenfold, to about 1,000 a year, and is predicted to reach 4,000 early next century." End quote. Italian researchers reported in the May issue of Genes, Chromosomes and Cancer, 2000, 28, 23-30. Quote, "More than a quarter of human giant cell tumors, GCTs, have DNA evidence of Simian Virus 40. Other studies reported that SV40T antigen, a viral protein, binds to human tumor suppressor proteins such as P53 and Rb. Research of Carbone, De Luca, Zen, suggesting a possible carcinogenic mechanism." Quote, "Additional testing for SV40 suggested that a large part of these antibodies may be antibodies to the human virus BK, and not to SV40. Because the DNA of the SV40 virus is nearly 70% identical to the BK virus, it is difficult to distinguish between antibodies of the two viruses. Most humans carry antibodies to BK in their blood, since the virus commonly infects humans as children. BK, however, is not associated with any disease in healthy people." End quote. Taken from Carbone, 1997. Gerber stated, quote, "To eliminate severe outbreaks of respiratory illness in basic training camps, the U.S. Army administered an inactivated adenovirus vaccine, grown in monkey kidney tissue, to entering service personnel in 1960 through 1961. Evidence is compelling that this vaccine was widely contaminated with live SV40. Adenovirus grows extremely poorly in monkey kidney tissue without the presence of SV40 as a helper virus." End quote. Concerning SV40, the IMUS Institute of Medicine concluded that 1. Quote, "The evidence is strong that SV40 is a transforming virus, and 2. The evidence is of moderate strength that SV40 exposure could lead to cancer in humans under natural conditions." From Carbone Study, 2002 and IMUS Institute of Medicine, 2002. Klein, et al., 2002 in Carbone, 2003. Oncology. Quote, "The presence of SV40 in human tumors has been convincingly demonstrated." End quote. We have gone even further. using SV40 to immunize against hepatitis B. We report here successful immunization using such an SV40-derived viral vector. Hepatitis B surface antigen, HBsAg, DNA was cloned downstream of two tandem SV40 early promoters to yield a T-antigen deficient SV40 derivative, SV, HBs. End quote from Kondo Research, 1998. This was a National Institute of Health solicitation for study found on the Internet on November 2004. The site was dated June 3, 2003. Quote, SV40 and non-Hodgkin's lymphoma, NHL, a case control study. End quote. SV40 was an inadvertent contaminant of poliovirus vaccines widely administered in 1955 through 1962 in the U.S. Of concern. SV40 causes a variety of tumors when injected into newborn laboratory animals, including brain tumors, mesotheliomas, osteosarcomas, and notably, NHL, non-Hodgkin's lymphoma. The possibility that SV40 infections, arising through vaccinations or other routes, cause a substantial fraction of NHLs was recently raised by two U.S. reports, describing the detection of SV40 DNA in 33 to 50% of NHLs. An SV40 VLPEIA was used in a study of NHL in Spain. In the first 2 years of the epidemic in San Francisco, between 50 and 60% of the earliest known AIDS cases were from persons involved in the hepatitis studies. Strong link between the U.S. outbreak of AIDS and hepatitis studies that were performed on gay males, starting in the late 1970s. Full-blown AIDS should have been evident many years earlier, before HIV was nearly so widespread. Thousands of infections would have been necessary to fuel the levels of HIV growth that were observed, during years in which no retroactive evidence of HIV exists. Taken from Internet site, http://www.bhc.edu/east-campus/AIDS/aidstesc.htm. The entire American population was subjected to the SV40 in either the original inoculations or from the shedding of the virus to others. Dr. Howard Ernovitz at the 8th Annual Houston Conference on AIDS cited significant evidence that, quote, human immunodeficiency virus type 1, HIV-1, is a monkey hybrid virus which was produced when 320,000 Africans were injected with poliovirus contaminated with live simian immunodeficiency virus, SIV. In the late 1950s, people who lived in Massachusetts and Illinois in the 1950s, and received identified lot numbers of the contaminated oral vaccine, are now contracting osteosarcoma bone tumors at a rate of 10 times more than those who received the vaccine free of the SV40. SV40 were identified in polio vaccines in Washington, Oregon, Wyoming, Utah, Minnesota, Iowa, Wisconsin, Illinois, Michigan, Pennsylvania, Washington, D.C., Maryland, Delaware, New York, Connecticut, Rhode Island, Massachusetts, Vermont and New Hampshire. Low levels of SV40 were found in California, Arizona, New Mexico, Colorado, Texas, Kansas, Nebraska, North Dakota, Missouri, Louisiana, Georgia, Tennessee, Kentucky, Ohio, and West Virginia. End quote. Polio vaccines in the other states show no SV40 present. In the USA, vaccine lots received by about 20 states are estimated to have contained 0.75 to 0.97 milliliters contaminated vaccine per child. Lots from about 15 states were thought to have contained 0.01 to 0.74 milliliters contaminated vaccine per child, and about 15 states were believed to have received lots that were free from SV40. By 1963, the estimated number of tainted polio vaccinations was estimated to be upwards of 98 million. Researchers from the Institute of Histology and General Embryology of the University of Ferrara, led by Dr. Fernando Martini, discovered SV40's presence in a variety of other tumors. They found the rhesus monkey virus in 83% of choroid plexus papillomas, in 73% of ependymomas, in 47% of astrocytomas, in 50% of glioblastomas, and in 14% of meningiomas. SV40 has appeared in 61% of all new cancer patients, patients even too young to have received the contaminated vaccine being administered 40 years ago. SV40 is believed to suppress the transcriptional properties of the tumor-suppressing p53 in humans through the SV40 large T antigen and SV40 small t antigen. p53 is responsible for initiating cell death, apoptosis, or cell cycle arrest when a cell is damaged. A mutated p53 gene may contribute to uncontrolled cellular proliferation, leading to a tumor. See Wikipedia. SV40. SV40 also has been found in 23% of blood samples and 45% of sperm fluids taken from normal individuals. Researchers have determined the SV40 virus can be transmitted sexually and through blood transfusions. SV40 DNA sequences have been detected in human pituitary gland tissue, peripheral blood mononuclear cells, and seminal fluids from healthy persons. Apparently, viral fragments combine easily with other viruses to produce hybrids called chimeras. 3 polyomavirus have been found in humans: Jamestown Canyon virus, JCV, BK virus, BKV, and Simian Virus 40, SV40. Prior to this, health officials were blaming AIDS on the habit of certain Africans to consume monkey flesh, as a result of the earlier contamination. It is estimated that 10 to 30 million people vaccinated in the United States from 1955 through early 1963 were inadvertently exposed to live SV40 virus. Taken from a report Simian Virus 40 and Human Cancer redistributed by the University of Bonn Medical Center. Hooper found, it is now apparent that the American, Belgian and French governments were all actively involved in the experimental polio vaccination trials that were conducted in colonial Africa in the late 1950s. There is now compelling evidence that the Belgian-American trials were responsible for the sparking of the AIDS pandemic in the Belgian Congo and Rwanda. The information about the French trials is more sparse, but is enough to indicate that they may well be linked to the 3 minor outbreaks of AIDS that occurred in France's former colonies of French West Africa and French Equatorial Africa. Given this, it is perhaps not surprising that at least some of these governments are now resorting to carefully coordinated disinformation campaigns. The documentary film titled Origins of AIDS was produced in November 2003 and viewed in the US on Channel 4, revealing witnesses testifying that they as nurses, Joseph Limbaya, Philip Elib, and Jacques Kanima have been a part of the live 400 chimpanzees production of the oral Salk vaccine in the Stanleyville, Kisangani, Congo. The Lindy camp was managed by Dr. Ghislaine Courtois, and the oral Salk was then distributed worldwide. No testing of the vaccine, for viruses, was performed on batches produced from 1956 through 1958. It was estimated 90 million children were vaccinated from these batches. The following are various research, abstracts and relevant opposing comments on the topic of Simian Virus 40. High-throughput testing of the SV40 large T antigen binding to cellular p53 identifies putative drugs for the treatment of SV40-related cancers. Carbone M., Rudzinski J., Becketta M. Loyola University Medical Center, Cardinal Bernardin Cancer Center, Department of Pathology, Maywood, Illinois 60153, USA. Virology 2003 October 25, 315, 2, 409-14. SV40 has been linked to some human malignancies, and the evidence that this virus plays a causative role in mesothelioma and brain tumors is mounting. The major SV40 oncoprotein is the large tumor antigen, termed TAG. A key TAG transforming activity is connected to its capability to bind and inactivate cellular p53. In this study we developed an effective, high-throughput, ELISA-based method to study TAG p53 interaction in vitro. This assay allowed us to screen a chemical library and to identify a chemical inhibitor of the TAG binding to p53. We propose that our in vitro assay is a useful method to identify molecules that may be used as therapeutic agents for the treatment of SV40-related human cancers." End quote. Two studies, DiRienzo et al. 2002, Emery et al. 2000. Examine mesothelioma tissue from a small number of patients in Turkey, where SV40-contaminated vaccines were not used, and found no SV40. The researchers also examined mesothelioma tissue from a small number of patients in the US and Italy, where SV40-contaminated polio vaccines were used, and found SV40 in some of the specimens. End quote. In 1999, Fisher and colleagues reported "increased rates of ependymomas, osteogenic sarcomas, other bone tumors, and mesotheliomas among people who were potentially exposed to SV40-contaminated polio vaccine." However, the number of cases in this study was too small to draw any statistically valid conclusions. End quote. In 1998, the National Cancer Institute published findings from a study, Strickler et al. 1998, revealed that, "after 30 years, there was no increased incidence of cancer in persons who may have received vaccine containing SV40. The study looked specifically for types of rare cancers that have been found to contain SV40 in recent cellular research, Carbone et al. 1994. And found no significant increased incidence compared with persons who had not received contaminated SV40 vaccine." End quote. Olin et al. 1998, conducted a long-term follow-up study of 700,000 people in Sweden who received polio vaccine potentially contaminated with SV40 in 1957 as school-aged children. Their results revealed no increased cancer incidence between persons who received vaccine containing SV40 and those who did not. End quote. Giesler, 1988, analyzed German National Cancer Registry data to compare the incidence of cancer in 885,783 persons born between 1959 through 1961 who received polio vaccine that may have been contaminated with SV40. It was compared it with 891,321 persons born between 1962 through 1964 who received polio vaccine free of SV40. The data demonstrated that persons who received polio vaccine possibly contaminated with SV40 did not develop more tumors within a 20-year period than did those who received polio vaccine that did not contain SV40. End quote. Mortimer, 1981, studied cancer deaths of 1,073 persons born between 1960 to 1962 who received oral poliovirus or inactivated poliovirus vaccine that contained SV40 when newborn. The follow-up study over 17 to 19 years revealed no increased number of deaths from cancer. In 2001, a 35-year follow-up study of this group was published. The study found no deaths in the group due to tumors of the type that have been associated with SV40. Follow-up study came from Carol Pankhurst, 2001. End quote. Formini et al. 1970 followed 1,000 persons who had received SV40 contaminated poliovirus vaccine within a few days after birth. The majority of these people received the SV40 contaminated oral vaccine at 8 years of age. No cancer deaths were identified in the exposed group. End quote. Formini et al. 1963 focused on a cohort of children aged 6–8 years who received inactivated poliovirus vaccine in 1955. A comparison was made based on whether children received vaccine with high, low or no detectable amount of SV40 contamination. Mortality rates from leukemia and all other cancers from 1950 to 1959 were compared across the three groups. No differences in cancer rates were found for this period. End quote. Two studies concerning maternal vaccination with SV40 contaminated vaccines and risk of cancer in offspring have been conducted. End quote. 1. Heenan et al. 1973 reported a higher incidence of neural malignancies in children born to mothers who received inactivated poliovirus during pregnancy. The prospective study of over 50,000 women who were pregnant between 1959 through 1965 identified 24 malignancies in their children during the first 4 years of life. The rate of malignancy was about twofold greater in children born to mothers immunized during pregnancy when compared with children born to unimmunized mothers or mothers who received influenza or OPV vaccines. Neural tumors accounted for most of the difference. End quote. 2. Farwell et al. 1979 found that of 15 cases of medulloblastoma in children born in Connecticut between 1956 through 1962, 10 were born to mothers exposed to SV40 contaminated polio vaccine while 5 were born to mothers unexposed. Interpretation of these results, however, is hampered by the low response rates and uncertain accuracy of vaccination histories by obstetricians. A follow-up study was conducted with Strickler et al. Blood tests can identify if a person has antibodies to SV40, but there was no test for SV40 commercially available at the time. Research laboratories are currently refining the techniques used to detect SV40. PCR, polymerase chain reaction, assays are currently in use to detect SV40 DNA segments. Because of inconsistent results between laboratories, there is a need to develop a standard PCR assay. Levine et al. 1998. End quote. On the 24th of July, 2002, an article titled SV40 in Vaccines was published in Atlanta. The U.S. Institute of Medicine, IM, was looking into whether polio vaccines contaminated with the Simian virus SV40 thought to inactivate P53NRB and given 40 years ago have caused a spike in the incidence of several cancers. As much as 2/3 of the oral Sabin and injected Salk polio vaccine supply given from 1955 through 1963 in the US, Australia, Canada, Germany and other countries was contaminated with SV40. According to a recent report in the Wall Street Journal. Since then, the incidence of certain rare cancers such as the brain cancer ependymoma has increased 25%, osteosarcomas are up 23%, and the incidence of mesothelioma, a lung cancer strongly linked to asbestos exposure, has soared 90%. The incidence of non-Hodgkin lymphoma, not a rare cancer, has increased by 82% since 1973. A total of 56,200 new cases and 24,000 deaths from non-Hodgkin lymphoma were reported in 2001. End quote. At the July 11 meeting of the IM's Immunization Safety Review Committee on the subject, Janet Butel of the Baylor College of Medicine, in Houston, Texas, presented data supporting the link between SV40 and non-Hodgkin lymphoma. In March, Butel and colleagues published a study that found genetic sequences from SV40 in 42% of the non-Hodgkin lymphomas they analyzed. 3 of those non-Hodgkin samples contained strains identical to that in the 1955 polio vaccine. End quote. Dr. Janet Butel told The Telegraph, quote, I feel strongly that research is warranted to determine how common human infections by SV40 may be, and what factors might predispose individuals to SV40-related tumors." End quote. Her study, published in the Journal of the National Cancer Institute, also suggested that, quote, "the monkey virus may be passing from those given the contaminated vaccine to their children, spreading the cancer risk still further." End quote. Quote, "Blood samples analyzed by Dr. Butel and her colleagues point to the steady spread of the cancer-causing virus in the human population, with 10% of those never exposed directly to the contaminated vaccine testing positive for SV40. Dr. Butel said, quote, "I believe SV40 is present in the human population today and is being spread among individuals by an unknown route." End quote. But Kirti Shah, of the Johns Hopkins Bloomberg School of Public Health, Baltimore, Maryland, argued at the meeting against the link, stating, quote, "suggesting that lab studies have been too inconsistent. The virologic and serologic data do not indicate that SV40 infection is widely disseminated in the community," one of Shah's slides read, "and it will not be possible to evaluate the etiologic link between SV40 and human tumors until the virus transmission is characterized." End quote. Epidemiologist Howard Strickler of Albert Einstein College of Medicine in the Bronx, New York, stated, quote, "Certainly, I think the evidence is there, that SV40 or fragments of SV40 are present in a wide variety of tumors." End quote. William Egan, Deputy Director of the Office of Vaccines Research and Review in the FDA's Center for Biologic Evaluation and Research, told The Scientist, quote, "But I don't think that anyone at this point can say whether SV40 is causing these tumors." End quote. Egan, quote, "And the question is, where is it coming from and how is this circulating in the human population now?" End quote. A conversation with Emma Hitt, Emma, at emmaschienenwriter.com, quote, "SV40 is a small double-stranded, circular DNA virus of rhesus monkey origin. It is closely related to JC and BK viruses of human origin. While BK virus may not cause human disease, JC virus can cause a very severe brain illness in HIV-infected patients. That is called progressive multifocal leukoencephalopathy or PML. SV40 is also distantly related to human papillomaviruses, some of which cause cervical cancers. SV40 infection is now widespread within the human population almost certainly as a result of polio vaccine produced in rhesus monkey kidney cells during the 1950s. A recent study showed infection in 23% of blood samples from normal individuals. The virus can also be detected in sperm fluid and is likely to be passed congenitally to future generations. Martini, et al. Research. End quote. Quote, "Other reports have shown SV40 in human brain tumors, such as the Berg-Sagil study, that SV40 has also been detected in a high proportion of human mesotheliomas, supported by Carbonette, al. Oncogene 9, page 1781 to 1790, 1994, and in bone tumors called osteogenic sarcomas, Carbonette, al. Oncogene 1996. End quote. Quote, "SV40 contamination was detected in the rhesus monkey kidney cells used to make polio vaccine as early as 1960. A federal employee, Dr. Bernice Eddy, decided on her own initiative to test extracts from the monkey kidney cells used to make polio vaccine for possible cancer-causing agents. She chose to use newborn hamsters since these animals develop tumors with the type of virus she and Sarah Stewart had discovered in mice and named polyomavirus. The inoculated hamsters developed tumors similar to those induced with polyomavirus. The results of Dr. Eddy's unauthorized testing was met with the same scorn and indifference as her earlier warnings that some of the initial lots of Salk vaccine retained live poliovirus. Dr. Eddy went around channels and disclosed her results in a 1990 scientific meeting. Her punishment was to be taken off polio vaccine safety testing and prohibited for over a year from submitting her work for publication. Dr. Eddy's findings did however leak out and were replicated by Drs. Sweet and Hillman at Merck. Initially, Hillman thought the SV40 problem would apply mainly to the live polio vaccines developed by Dr. Sabin and used extensively in the Soviet Union. He even suggested that the Russians would be no threat at the upcoming 1962 Olympics because they would be dragging with tumors. The Health Century by Edward Shorter. Unfortunately, it was soon realized that the polio vaccine developed by Dr. Salk was the more dangerous because the 1 in 4,000 dilution of formaldehyde, which barely inactivated the polio virus, did not fully inactivate SV40. Because the Salk vaccine was injected through the skin, it allowed the SV40 a better chance to infect. End quote. Dr. Hillman further stated, quote, "Use of tissues of wild-caught animals is just asking for trouble because of the lack of control and the known high probability for viral contamination. Monkeys are too expensive to be grown in specific pathogen-free colonies and, hence, the simple solution to the monkey problem is to eliminate the monkey and also the tissues of wild-caught animals, and certainly monkeys, are commonly infected with wild viruses. The simplest way to solve the monkey problem is to eliminate the monkeys, and this is being done using diploid cells." End quote. Quote, "Monkey tissues came into use by historic decisions to use monkey kidney to make polio vaccine. There is no need to continue using monkeys when acceptable alternatives are available." End quote. Quote, "The main result of finding SV40 virus in rhesus monkeys was simply to switch polio vaccine production to African green monkeys. Existing stocks of SV40 polio vaccines were not withdrawn from the market, nor was any effort made to suspend the military's use of adenoviral vaccines, also known to contain SV40. Short-term follow-up studies on some of the children exposed to SV40, to see if brain tumors had developed, satisfied those in charge of public safety, that no harm had been done." End quote. Quote, "While the African green monkey is generally free of SV40 virus, it does carry a number of viruses, including Simian Virus, SCMV. As an aside, rhesus and not African green monkeys are used for testing the virulence of live polio vaccines, because even the control African green monkeys often show evidence of neural infection." End quote. Quote, Dr. Ratner, a public health official whose concerns about polio vaccine safety had been largely ignored, have been sent to investigators studying SV40 to show how easy it is to detect contaminants using the PCR assay. This assay was not available in the 1950s, but now that it is, there is no excuse for not using it on polio and other vaccines to exclude contaminants including SCMV and related stealth viruses. For information on SV40 testing, please refer to the clinical laboratory http://www.ccid.org/adviruses/SV40.htm 2002. End quote. Dr. Joseph Merkeler's comment, quote, "Lancet showing that half of the 55,000 cases of non-Hodgkin lymphoma in the U.S. are likely related to contaminated polio vaccine." End quote. Quote, "The researchers believe that the contaminated vaccine batches were cleared up in 1963. To date, the scientific literature and research examining SV40 and cancer-related diseases has been based upon an assumption that SV40 was not present in any poliovirus vaccine administered in the United States and was removed from the killed polio vaccine by 1963." End quote. Vilches, et al. Stated, quote, "There are no commercial tests to evaluate SV40 infection. Serologic assays such as ELISA for SV40 have a low sensitivity. In addition, A recent FDA panel concluded that none of the current ELISA tests for SV40 are reliable for research or diagnostics. While the serum neutralizing antibody test is the recognized gold standard serologic test for SV40, it has low sensitivity and requires great labor. Therefore, molecular assays such as polymerase chain reaction, qualitative and quantitative, have been used to study the relation of SV40 infections and human malignancies. These tests are currently for research purposes, but different laboratories, including ours, are working to establish them for commercial and diagnostic use. Indeed, this is one of the recommendations of the Institute of Medicine for studies of SV40 in humans. We hope they will be available to patients in the near future. Doctors estimate that the virus was injected into tens of millions during mass vaccination campaigns before being detected and screened out in 1963. Those born between 1941 and 1961 are thought to be most at risk of having been infected. End quote. The Telegraph Group Limited 1999 has learned that quote, scientists in Britain have joined an international effort to confirm the findings. According to Prof. Gordon McVie, the Director General of the Cancer Research Campaign, researchers have so far uncovered evidence linking SV40 to a number of cancers, including brain tumors and bone cancer. He said, "I've a feeling that the virus might be implicated in more, such as non-Hodgkin's lymphoma and prostate cancer." End quote. Robin Weiss, a professor of viral oncology at University College, London, said quote, that SV40 is widely used in laboratories and could easily contaminate tumor samples, fooling the ultra-sensitive tests used to detect the virus. Robin Weiss, a professor of viral oncology at University College, London, said quote, that SV40 is widely used in laboratories and could easily contaminate tumor samples, fooling the ultra-sensitive tests used to detect the virus. Professor Weiss said, quote, "Many of the positive results are probably false positives. We've looked at mesothelioma and did find it in some cases, but then we got bogged down in whether they were due to contamination or not." End quote. However, Dr. Berit Jassani, a leading expert on SV40 and mesotheliomas at the University of Wales College of Medicine, Cardiff, said that quote, "new reliability tests rule out contamination as a possible explanation." He said, quote, "There is absolutely no question of laboratory contamination being to blame." End quote. Dr. Jassani said he had quote, "little doubt that the mass polio vaccination campaigns were to blame for SV40 entering the human population, suggested that many might be treated by a vaccine that attacked SV40." End quote. Quote, "In 1997, an international SV40 working group, which included the majority of laboratories studying SV40 in human tissues, was formed. 9 laboratories from the working group agreed to participate in a study, funded and organized by NCI. Each lab group was given 25 paired duplicate samples of human mesotheliomas, a single set of 25 normal lung tissue samples, and positive and negative control samples. All the samples were masked, prepared and labeled so that the human tumors and controls could not be distinguished. Each laboratory used one or more assays for detecting SV40, many of which had been used to detect SV40 previously. The results show that none of the mesothelioma specimens was consistently positive for SV40 across all laboratories." End quote. A review of 1960s Danish records identified widespread SV40 contamination of previously utilized Danish poliovirus vaccine, which, unlike in the US, was grown in pooled kidney tissue from dozens of monkeys. A further strength of this study by Engels, et al. 2003 was the high quality of Denmark's nationwide data on cancer incidence, which go back to 1943. Overall cancer incidence was actually lower in SV40 exposed individuals for those exposed as infants or children, respectively, compared with those unexposed. no increased risk was seen for specific outcomes, including mesothelioma, bone tumors, brain tumors, and non-Hodgkin's lymphoma, NHL. A brain tumors study in northern India, involving a population uniquely exposed to SV40 people living in northern India, was published recently. Engels, et al. 2002. Investigators did not find SV40 in any of the tumors. Researchers found no increased antibody levels to SV40 detected in lymphoma cases versus the controls. Overall, SV40 antibody levels were low in both cases and controls. The Institute of Medicine, IOM, of the National Academy of Sciences issued a report in October 2002, the Immunization Safety Review, which concluded that scientific evidence is inadequate to accept or reject a causal relationship between SV40 containing polio vaccines and cancer. Page 11. Executive Summary. Biological evidences of moderate strength that SV40 exposure could lead to cancer in humans under natural conditions, and that biological evidences of moderate strength that SV40 exposure from the polio vaccine is related to SV40 infection in humans. Notice the admittance of exposure in 2002. Because of the widespread exposure to SV40 through contaminated vaccines, the question of whether SV40 causes some human cancers has substantial public health implications. Concerning SV40, the IOM concluded that 1. The evidence is strong that SV40 is a transforming virus, and 2. The evidence is of moderate strength that SV40 exposure could lead to cancer in humans under natural conditions. IOM, 2002. Similar conclusions were reached at an international consensus meeting on SV40 and human tumors held at the University of Chicago in 2001. Klein and Cross, who chaired a final panel that reviewed all the published evidence linking SV40 to human tumors, and stated that the presence of SV40 in human tumors has been convincingly demonstrated. Klein, et al. 2002. 3 independent scientific panels have all agreed that there is compelling evidence that SV40 is present in some human cancers and that SV40 could contribute to the pathogenesis of some of them. As cited in Carbone, in Oncogene, 2003, a possible human polyoma virus workshop. Monday, 27th of January, 1997. Afternoon session. The workshop took place in the Natcher Auditorium, National Institutes of Health, Bethesda, Maryland, at 8:30 a.m. Catherine C. Zune, Director, CBER, presiding, Dr. Patrick Holin, Swedish Institute for Infectious Disease Control, stated at the workshop, Swedish investigators were informed already in 1959 about the problems of SV40 in the U.S. The Mazzoni study, 2020. Conclusions. In this investigation, polyoma virus, termed PYV, vertical transmission was revealed by detecting PYV DNA sequences and IgG antibodies in samples from females, and their offspring suggested a potential risk of diseases in newborns. The mother-to-child transmission of oncogenic polyoma virus SV40 does occur. In 2015, Kluber, et al. Study supported evidence of SV40 urinary shedding. Carbone, 2020, stated, Together this evidence suggests that SV40 may contribute to the development of mesotheliomas in humans. However, epidemiological evidence to support this hypothesis is lacking. This chapter also summarizes the introduction of SV40, a monkey virus, into the human population as an unrecognized contaminant of early polio vaccines. In addition to mesotheliomas, SV40 now is linked with brain cancers, osteosarcomas, and lymphomas in humans. End quote. Notice the 15-year gap in rigorous research from 2000 to 2015. There is little justification as to why. The end.